Hi everyone, I am Dr. SHAGIRTHA, Assistant Professor, Department of Biochemistry, St.Joseph College of Arts and Science, Cuddalore. In this video, we are going to talk about carbohydrate metabolism and especially the cellular respiration. So, we will look out the different stages of cellular respiration. We think about cellular respiration starts with glycolysis, and then the process goes the modification of pyruvate and acetyl-CoA, that is done by pyruvate dehydrogenase complex. And the third stage, the process known as Krebs cycle. And the fourth stage, the process known as electron transport chain to generate energy. Now, we look out the overview of cellular respiration. It starts with glycolysis. The idea of cellular respiration is very simple, that we have eat food and the fragment of food after the digestion reaches our cells. And inside the cell, all those food particles are broken down into the macromolecules. That carbohydrate, lipids and proteins are major macromolecules produced after the digestion. So the idea of cellular respiration is to utilize all the macromolecules to produce energy inside the cell. They have different metabolic pathway. The carbohydrate, protein, lipids have separate metabolic pathways. Now, we are going to talk about carbohydrate metabolic pathway. This carbohydrate metabolism starts with glycolysis. Metabolism refers to the chemical process that takes place as your body converts food and drinks into energy. It is a complex process that combines calories and oxygen to create and release energy. Metabolism is a total amount of biochemical reaction involved in maintaining the living condition of the cell in an organism. All living organisms require energy for different essential processes and are producing new organic substances. The metabolic process helps in growth and reproduction and help in maintaining the structure of living organisms. The organism responds to the surrounding environment due to the metabolic activities. All the chemical reaction occurring in the living organism, from digestion to transportation of the substance from cell to cell, require energy. These are two types of metabolic process. They are catabolism and anabolism. Catabolism is a process is mainly involved in breaking down larger organic molecule into smaller molecules. This metabolic process releases energy. And the another one is anabolism. The anabolism is the process mainly involved in building up or synthesizing compound from simpler substance required by the cells. This metabolic process requires and store energies. Next one, carbohydrate in metabolism. Carbohydrates are supplied in three forms: starch, sugar and cellulose. Starch and sugar are the major form of energy for humans. Metabolism of carbohydrate and sugar help in the production of glucose. Next, the protein in metabolism. Proteins are important for building tissues that help in maintaining the structure of cells and its function, the formation of hemoglobin and the several other body functions. The amino acids of protein are beneficial for nutrients. Few amino acids are not synthesized by the body and are taken in from the food we eat. The amino acids include lysine, tryptophan, methionine, isoleucine, leucine, phenylalanine, valine and threonine. Now, we discussed how to increase the metabolism. First one, to be fit and healthy, we need to avoid more calories intake and lose extra pounds. We eat to deliver energy for our body to perform its function. Next, having Proper breakfast boosts up the body's metabolism and keep us energetic throughout the day. Skipping morning breakfast are most likely to have poor metabolic energy. Next one, the caffeine. Caffeine stimulates the central nervous system and can activate our metabolism rate by 5 to 8%. According to some researchers, fiber can help to burning fat by 30%. People who include more fiber in their diet remain fit and healthy. Next one, the including more organic foods like peppers, apple, lettuce, grapes can boost up the metabolism rate in our body. This is the functions of metabolism. Mainly breathing, circulating blood, digestion of food, growing and repairing cells, managing hormones level and regulating body temperature are the main functions of metabolism. Now we discussed about glycolysis. Glycolysis is the process in which glucose is broken down to produce energy. It produces two molecules of pyruvate, ATP, NADH and water. The process takes place in the cytoplasm of the cell and does not require oxygen. It occurs in both aerobic and anaerobic organisms. This glycolysis is defined as sequence of reaction converting glucose to pyruvate or lactate with the production of ATP. Glycolysis takes place in all the cells of the body. The enzymes of this pathway are present in cytosomal fraction of the cells. Glycolysis occurs in the absence of oxygen or in the presence of oxygen. Lactate is the end product under anaerobic condition. In the aerobic condition, pyruvate is formed, which is then oxidized to carbon dioxide and water. Glycolysis is the major pathway for ATP synthesis in tissues lacking mitochondria, example erythrocytes, lens, etc. And glycolysis is very essential for brain, which is depend on glucose for energy. The glucose in brain has to undergo glycolysis before it is oxidized to carbon dioxide and water. The reactions of glycolysis divided into three distinct phases: energy investment phase, splitting phase and energy generation phase. Now first we discussed about energy investment phase. This is the glucose. The glucose is phosphorylated to glucose 6-phosphate by hexokinase or glucokinase. This is an irreversible reaction depend on ATP and magnesium. The enzyme hexokinase is present in almost all the tissues. It catalyzes the phosphorylation of various hexoses and it's inhibited by glucose 6-phosphate. Hexokinase present in liver catalyze the phosphorylation of only glucose and not inhibited by glucose 6-phosphate. Glucose 6-phosphate is impermeable to cell membrane. It is a central molecule with varieties of metabolic fats like glycolysis, glycogenesis, gluconeogenesis and pentose phosphate pathways. This glucose 6-phosphate undergoes isomerization to give fructose 6-phosphate in the presence of enzyme phosphohexose isomerase and magnesium. This fructose 6-phosphate is phosphorylated to fructose 1,6-bisphosphate by phosphofructokinase. This is an irreversible and regulatory steps in glycolysis. Next one is a splitting phase. The 6-carbon fructose 1,6-bisphosphate is split into two 3-carbon components like glyceraldehyde 3-phosphate and dihydroxyacetone phosphate by the enzyme aldolase. The enzyme phosphotriose isomerase catalyzes the reversible interconversion of glyceraldehyde 3-phosphate and dihydroxyacetone phosphate. Thus, 2 molecules of glyceraldehyde 3-phosphate are obtained from 1 molecules of glucose. Next one is the energy generation phase. Glyceraldehyde 3-phosphate dehydrogenase converts glyceraldehyde 3-phosphate to 1,3-bisphosphoglycerate. This step is very important and it is involved in the formation of NADH and H+ and high energy compound 1,3-bisphosphoglycerate. Iodoacetate and arsenate inhibit the enzyme glyceraldehyde 3-phosphate dehydrogenase. In aerobic condition, NADH passes through the electron transport chain and 6 ATP are synthesized by oxidative phosphorylation. The enzyme phosphoglycerate kinase acts on 1,3-bisphosphoglycerate and resulting in the synthesis of ATP and formation of 3-phosphoglycerate. This step is a good example of substrate level phosphorylation. Phosphoglycerate kinase reaction is irreversible and rare example among the kinase reaction. This 3-phosphoglycerate is converted into 2-phosphoglycerate by phosphoglycerate mutase. This is an isomerization reaction. The high energy compound phosphoenol pyruvate is generated from 2-phosphoglycerate by the enzyme enolase. This enzyme requires magnesium and it is inhibited by fluoride. For blood glucose estimation in the laboratory, fluoride is added to the blood to prevent glycolysis by the cells so that blood glucose is correctly estimated. The enzyme pyruvate kinase catalyzes the transfer of high energy phosphate from the phosphoenol pyruvate to ADP, leading the formation of ATP. This step is also a substrate level phosphorylation. This reaction is irreversible one. Finally, the pyruvate, enol form of pyruvate is converted into keto form of pyruvate spontaneously. And finally, the pyruvate is converted into L-lactate by the enzyme lactate dehydrogenase. This is the factors affecting metabolism. The factors which are affecting the metabolism are muscle mass, age, sex, genes, physical activity and smoking. The first one is a muscle mass. It takes more energy to build and maintain muscle than fat. People with more muscle mass often have faster metabolism that burn more calories. Next one is the age. You lose muscle as you get older, which slows down the metabolism. Next one, sex. Males tend to have faster metabolism than females. They have more muscle mass, larger bones and less body fat. Genes. This is very important one. The genes you inherit from your parent play a role in your muscle size and ability to build muscle mass. Physical activity is very important for all the peoples. Walking, chasing after your kids, playing tennis and other forms of exercise cause your body to burn more calories than being sedentary lifestyle. Smoking is a very important factors affecting the metabolism. Nicotine speeds up your metabolism so you burn more calories. This is one reason people who quit smoking may put on weight. But the health consequences of smoking lead to cancer, high blood pressure, coronary artery disease. For overweight, any benefit you might get from burning a few extra calories. So avoid smoking is very important. So these are the factors which affecting the metabolism mainly by muscle mass, age, sex, genes, physical activity and smoking. So we avoid smoking is very important to boost up our metabolism. So thank you.